Thank you very much, Mr. Machila, for your wonderful welcome. So as you have said, today I'll be talking about electrochemical biosensor systems for diagnostics and environmental analysis. When you were addressing me today, you have mentioned that I am the thematic area leader of the applied electrochemistry. And the team members of the applied electrochemistry are myself, Dr. Sikosana, Dr. Matito, and Dr. Hlongwa. So basically, our group specializes in the synthesis and characterization and application of nanomaterials in various niche area themes. I have here our niche area themes that we have in the applied electrochemistry. We have environmental water sensors and electrocatalysis. So after the synthesis of these materials and characterization of the materials, because it is very important to monitor the sizes and the shapes before we apply them in environmental water sensors and electrocatalysis. So the people who are involved in this theme are myself and Dr. Sikosana. We have another thing, which is electrochemical energy materials. So the people who are involved in this niche area theme are Dr. Jack Matito and Dr. Ntutuko Hlongwa. So because we are a team, we needed to come up with a niche area that will combine all the expertise in our thematic area. So remember, I've said that we are going to be synthesizing various nanomaterials and then be applying them in these niche areas. But then how about we come up with another niche that is going to be to involve all the team members in our group? So we came up with electrochemical water desalination. And when it comes to the synthesis of these materials, we use various synthesis routes and apply those in electrocatalysis. I will be explaining more about electrocatalysis, sensors, and biosensors. Now, the content of my presentation will cover two parts. The first part will be just the description about biosensor or the principles of biosensor systems. And then I'll go into details now talking about the specific biosensor types, which will involve enzymatic biosensors, genosensors, and aptasensors. And then the second part will be, because my topic is about diagnostics part and water analysis. So the second part will involve the sensor or biosensor systems for endocrine disrupting compounds in water. And then I will talk more about our ongoing projects, which are actually on COVID-19 in wastewater and some pharmaceuticals in water bodies. What is a biosensor? This is a simpler description of a biosensor system. So it is an analytical device that is used for the detection of an analyte. An analyte can be an ARV drug, it can be a water or pharmaceutical contaminant in water. So when it comes to the biosensor means that you have a biological component that will be linked onto your materials and your electrode. So what we do with this biosensor system is that we are detecting, that means we are checking how much of these contaminants are present in water. Or if we are doing diagnostic biosensor, we're checking the amount of these drugs in water, in the body. So now how do we develop these biosensor systems? I was talking about biological materials there. So these biological materials can be also known as bioreceptors. So a bioreceptor can be an enzyme. I'll talk more about these things as I go through my presentation. An antibody, microorganism, and CDL. That is a bioreceptor. So you will have an analyte, a bioreceptor, you have a transducer. So these transducers are two. One is an electroactive substance, and the second one is the electrode. So an electroactive substance means that you have to use nanomaterials that are electroactive, because after those synthesis, you monitor these nanomaterials using optical properties, electrochemical properties, so that you can be able to use them in biosensor development. Now, when it comes to the development of the sensor or biosensor design, we use various electrode systems. So this slide is showing you various examples of electrodes that are used in the development of the biosensor systems. So you can use a classic carbon electrode. So normally, a classic carbon electrode has a diameter of 3 millimeters. So what happens is that after the synthesis and characterization of the nanomaterials, you can drop cast or electropolymerize your nanomaterials on the electrode surface using a classic carbon electrode. Another one is the screen printed carbon electrode. So it depends on the material that you're using, and then you can employ a variety of these electrodes. Now, the biosensor development normally uses 3 electrode system. So your working electrode can be the classic carbon electrode, it can be the screen printed carbon electrode, or an FTO electrode. Then you will have your reference electrode that was responsible for the potential change. Because when you are developing these sensors, you apply a voltage, a certain voltage, which is the potential window. You select a potential window and apply a voltage and get the current. So the current change is the counter electrode is responsible for the current change in the electrochemical cell. Now, I was mentioning something in the second slide there about the electroactive substance. So in this case, I just showed a few examples of electroactive materials. So these electroactive materials can be polymers, functionalized quantum dots, nanoparticles, and nanocomposite materials. So what is the importance of these materials before the fabrication or by using these materials for the fabrication of the biosensors? It's because of their properties. So if we are claiming that these materials have good optical and electrochemical properties, we need to synthesize and test them so that we can employ them further for our biosensor development. So now when it comes to the preparation of these nanoparticles or quantum dots, we normally use functional, the capping agent or the stabilizing agent to make them stable and to make them biocompatible. What do I mean by biocompatible? To make them suitable for attachment of another group on top of those quantum dots. Normally we use the capping agents that have thiol groups, amino groups, and carboxylic group, because those groups are suitable for attracting other or biological material to bind on top of those quantum dot functionalized. Well, now when it comes to the biorecognition or a bioreceptor that I was talking about in the previous slides. So I've mentioned enzymes, I've mentioned antibodies. So how do these bind onto the functionalized electrode? We have our electrode, we've spoken about the nanomaterials functionalized, and now we are talking about a bioreceptor. So in most cases, if you are using an antibody, for an example, you have to actually block because there are some non-specific sites. So there are a variety of blocking agents that can be used to block the non-specific sites when developing a biosensor system. So in this case, most of the people are using bovine serum albumin as a blocking agent. So now if we are moving to the enzyme, we are looking at the heme center, but I'm going to be talking about their details in the next few slides. We are looking at the active site. That active site is responsible for binding the analyte. What are these enzymes? I'll first talk about enzyme-based biosensors. So these enzymes are used to catalyze carcinogens, drugs, environmental pollutants, and hormones. I've spoken about the heme center or an active site. So in this case, we have our active site. It is also known as the heme center. So the right-hand side is showing the mechanism of how the drugs bind, or these hormones bind onto the active site of the enzyme. So what happens is that you have your analyte and you include it in your cell after developing your biosensor system. So once you include or inject your analyte, that analyte binds onto the heme center. You see, in this case, the heme center has ion 3, and this RH means that you have included your analyte, and RH is an analyte in this case. So once you have that analyte here in solution, it automatically converts the ion center, the heme center, from ion 3+ to ion 2+. That means that you are reducing 1 electron. The second step is that when you are developing an enzyme-based biosensor, you require oxygen, so that this oxygen binds onto the heme center as well. Now, once you include that oxygen onto the heme, you can see that the oxygen, yes, binds onto the heme center as well. You have your analyte and you have your oxygen binding, and then you remove one of the oxygen there. So the reason why now you have an active metabolite or a byproduct, it means that you are breaking down this oxygen. You are doing a mechanism called monooxygenation. So you are breaking down this double oxygen into monooxygenation. As you can see here, your byproduct is released after forming a monooxygenation process. I have spoken about the electrodes that are used in the fabrication of the biosensor systems. And now there are various types of biosensors one can develop. We can produce an optical biosensor, which means that you can use a UV-Vis or fluorescence to have your optical biosensor systems. You can use piezo electrochemical biosensor systems, but today the focus is on electrochemical biosensors. Now this is the design, a simple design of the biosensor system. So in this case, we developed an ARV biosensor system. So we were using gold electrode as our working electrode, and then we modified the L-cysteine or L-cysteamine. So there are reasons for including L-cysteamine or L-cysteine onto the gold electrode surface. The reason is that thiol groups, L-cysteine has a thiol group, as you can see here. So the thiol group has a high affinity for gold electrode. So once we have this thiol group on the surface of the electrode, we have another group that is free for attachment of another layer. I have stated that we synthesize and functionalize nanomaterials, quantum dots, because we want them to be biocompatible or to be able to bind with another layer. So in this case, we have a carboxylic group and we have an amino group. So looking at our functionalized quantum dots, we also have carboxylic group that is free for attachment of another layer. So how do we now combine or attach the quantum dots onto the L16 modified electrode? So we need to introduce now the cross-linking agent. The cross-linking agents are used to activate these functional groups. So normally we use N-hydroxycyanamide, which is used to activate the amino group, and we use another cross-linking agent, which is EDC, which is actually used to activate the carboxylic group. So in this case, once we introduce the cross-linking agent, we automatically form a strong mi bond because this is activated and this is activated, and we form our another layer. So it's layer by layer approach. Okay, now once we have these ones, because you see we have activated this group, so this group is still free because you see we have attached the carboxylic group onto the amino group, and some of these functional groups from the quantum dots are still activated and free for the attachment of the enzymes. So enzymes also have the amino group and carboxylic group. So we can actually now take these quantum dots that are functionalized and attach the enzymes onto the quantum dots modified electrode to form our biosensor system. So what is the approach for understanding the biosensor system? Because we were putting layer by layer. We have L16 or cysteamine on the electrode. We included another layer on top of that, and then an enzyme. So we have 3 layers on the electrode. So one of the things that one should look at is that we need to now characterize layer by layer. The first layer we have is the L16. So what are the signatures of the L16 compared to that of the bare electrode? Because we need to understand our biosensor before applying it. So now if you are looking at the bare, this is where we were using gold electrode. So if you are comparing now the cysteamine onto the bare electrode, we can see that, of course, we have formed, thiols have high affinity. Although the signatures did not change that much, but we could see some enhancement in terms of the signatures for the L16. So now it means that once you have something on the electrode, you actually have some electron transfer. So another important point is that we did our L16 moving on to the quantum dots on their own, and we are comparing these quantum dots with the bare electrode before combining the enzyme now on top. So looking at our bare, the bare is the black line still, and then the red is the quantum dots. So we can see that the quantum dots has several peaks, redox peaks. We call them redox. It's oxidation and reduction. So the quantum dots exhibited several redox peaks, which are attributed to the palladium, tellurium, and the stabilizing agent. Now moving on to the right-hand side, because we are doing these things layer by layer, the red line in this case is the enzyme on its own on the electrode surface. So we can see that the enzyme exhibited two oxidation and 2 reduction. So how do we now compare our final sensor with the enzyme? Because the enzyme is responsible for this binding. We are looking at the heme center. So what are the signatures that we need to monitor when it comes to the sensor on its own before the addition of the analyte? So we need to look at the signatures that are attributed to the sensor or the biosensor. So we can still see that these signatures, most of the signatures that are in our sensor platform are still coming from the quantum dots. So we can see that if you're looking at the enzyme on its own, this is the redox, first redox, second redox. We can see if you're looking at the reduction, because we were saying that in the mechanism, we are doing 2 electron reduction. One is after the binding. The second one is after the addition of the oxygen. So in this case, we are looking at the 2 reduction peaks. We have a reduction peak around 0.1, minus 0.1, and have another one at minus 0.6. So these are the signatures for the heme center, because we were saying we are changing, moving the state ion 3 plus to ion 2 plus. So this is what is happening now after changing the states. And this one is actually responsible for the monooxygenation, because monooxygenation happens at the longer or more negative potential. So the peak that we are monitoring in this case is A1, because you see even here, although there is a slight shift that is happening here, we can still see that yes, our enzyme is here. Okay. As I was saying that we are monitoring this region. So why is this region, why is this graph different from the other graphs? This graph is different because now we are detecting our ARV drug. So in this case, we are adding various or different concentrations of the ARV drug. So you can see that we have added more than 10, more than 15 concentration. It is very important. The reason why we need to add from 10 up to 20 is because we need to get a longer linear range, and also the sensitivity and our limit of detection from that linear progression. So we can see that as we are increasing the concentration, as we are increasing the addition of the concentration, these peaks are also increasing. But this one actually is increasing with increasing concentration. You see up to minus 32, the current of 32. So how do we now calculate, how do we come up with our linear curve? Knowing that the cyclic voltammetry, it has a background current. So when you are using this type of technique, one, we are monitoring the redox signatures. The redox means you are monitoring the oxidation site, which is the forward scan. So you are scanning this way. The forward scan means that you are moving to the more positive potential. The reverse scan, you are moving to the more negative potential. So the most important thing in this case here is that we need to subtract the background from the sample. So before we go to our linear regression, we need to monitor. We need to be subtracting the first addition from the background, second addition to the background, so that we understand our linear progression. We get a proper linear regression. So after the subtractions, we plotted our linear curve. And it is important to note that when you are developing these biosensor systems, you need to do the experiments 3 times so that you can get a proper linear regression, and also include the error bars in this case. So we can see that we have obtained a sensitivity of 0,0218 and a longer dynamic linear range because we have added more than 10 concentrations. So it is very important when you're coming up with your biosensor system, you use like a variety of concentrations. And you go lower because most of the time when you are starting your concentration higher, you might reach a saturation point easily. Now, another important factor when developing the biosensors is that after doing the detection part, you need to check the specificity, meaning that you need to check if your sensor is just for the ARV drug or whatever. So in this case, we have included paracetamol because, you know, we get sick. People who are taking ARV drugs can actually take Panado as well. So we needed to see what will be the interference if we include the Panado in our sensor system. So because we are monitoring our signature in this case, the Panado did not interfere with our sensor system. So the biosensor is specific for the ARV drug. After doing the interference study, you can move on and continue with real sample studies. So in this case, we employed human serum. So, I mean, we did a stability study, sorry, on the right-hand side, because we need to, after doing the interference, because you need to prove that you have developed a sensor for your ARV drug, and then you need to check the stability, how stable it is, because most of the time these enzymes denature. So we monitored the stability of the enzyme, and we could see that the our biosensor was stable. Okay. So once you have your sensor, it is very important to compare the performance of the biosensor system with other technologies that are already in use. So in this case, we can see that the looking at the dynamic linear range and the limit of detection, the performance of our biosensor system was better. And you can see that even the limit of detection is lower than all of these biosensors that are already developed. So biosensors are the way to go. So when it comes to the real sample experiments, we employed human serum, and we took 3 samples. So it is very important now to check the added concentration, and then you check the found concentration after those additions. You calculate the recovery study, and you calculate their relative standard deviation. That is very important. And we have published this work in Electrocatalysis. Now, another type of the biosensor is the aptamer sensor. So aptamers are single-stranded nucleic acid molecules selected against target compounds to which they bind with high affinity accompanied by conformational changes. So why are these aptamers very important? Because of their high selectivity. And they normally developed for specific binding. And compared to other biomolecules, they are regarded to have longer shelf life, and they do not denature that easily. So what is the procedure for synthesizing or coming up with these aptamers? So one of the ways of synthesizing these aptamers is that you have your target analyte. You can have your target analyte, and you incubate this target single-stranded DNA library. So once you incubate this analyte with this, you remove the unbound sequences. and you continue with the positive side, because it is very important that we use only the positive side when we are using these aptasensors. So there are various methods for modifying these aptamers. So if you're buying these aptamers from Sigma, you'll see that most of the time you can have a thiol 5 thiol, or you can have bio-T. So it's up to you. It depends on what your application is all about, so that you can buy a specific aptamer for your specific work. So there are various methods for using these aptamers. There is a direct capturing. I'll be talking about that in the next slide. So if we're talking about the direct capturing, remember we have our sensor system, we have our nanomaterials and the aptamer. And in this case, because we were talking about specific sites, we need to block the non-specific sites. So we use the MCH as a blocking agent. You see, this is the blocking agent. So it is important to block the non-specific site so that it does not interfere with the signals. So once you have the direct capturing, it means that you will have your aptamer, and because the sites that are opened here are specific, so the analyte will be grabbed, will be here, will be captured, because you are focusing on the direct capturing of the analyte. Now, when it comes to the results, I have spoken about cyclic voltammetry, and that instrument is used for the redox properties. We have now impedance spectroscopy. This technique is used to measure the phase angle. So we will measure the phase shift, and we also measure the resistance to charge transfer. So in this case, once we have now, this is our aptasensor. So we included, or we added various concentrations of the TB biomarker, which is called the interferon gamma. So we were adding the different concentration of interferon gamma. As we were adding, we noticed that these signatures, or the RCT, was increasing with increasing concentration. Also, these results are complementing the results that we are obtaining on the right-hand side, that as we are increasing the concentration, the phase angle is also shifting. And then similar story here, we take the readings from the maximum. Here, we'll be taking from the maximum frequency. We take our readings from here and plot our linear regression plot, where we are monitoring the limit of detection, sensitivity, and all that. When it comes to the DNA is similar to the aptamers, because we're looking at the stable double-stranded DNA. So if you are looking at the double-stranded, you'll see you'll have your AT-TA, you'll have that. But if you are looking at this one, that is a strong hybridization. If it is a weak hybridization, the GT will be mismatched. So we need to focus on the strong hybridization when we are doing developing our biosensor system. So the method is still the same, because even for the aptamers, we were saying that we have 5 thiol, 5 bio-T, depending on your application. In this case, we have thiol DNA. So thiol means that your DNA will have a sulfur group there present. So this is the actual structure of what we have in terms of the DNA. And the procedure now for attaching the other materials onto the electrode is the same, because we were talking about the cross-linking agent to actually activate the groups that are present in your biosensor system. So these are the type of results we obtained from the DNA-based sensor with the limit of detection calculated on the right-hand side. So the procedure is the same, but then the biomolecule will be different. Also, the signature might change. Some of the signatures, you can monitor the signature in the reduction side, or you can monitor the signature on the oxidation side, depending on what you are using as the biomolecule. Now, part 2 involves the biosensor or sensor systems for water management. So we are still using the aptamers, but this aptamer in this case is used for cytocysteine. So the development in this case, remember when I was presenting the first part, I was focusing on the polymers and nanoparticles and quantum dots. We have here polymers and silver nanoparticles. So it's the combination, it's a nanocomposite because we are combining two materials there to form a nanocomposite. So this is just the type of the signatures we obtained, because as I was saying that if you are looking at the phase angle, we are looking at the maximum frequency in this case to see what type of signatures or what type of the linear range you are going to obtain. So we have both plot, which is measuring, Nyquist plot, measuring the RCT, and we have the both plot, the phase angle, which is focusing more on the phase. So most of the time, if you are doing electrocatalysis or sensor without any biomolecule, you can actually monitor or use the phase angle to check if your nanomaterial, your quantum dots, or your polymer is a semiconducting material. And we have also checked the interferences. We have found that all these other contaminants did not interfere with our analyte. And we have published this paper this year. Now, I was speaking about sensor or electrocatalysis. When we are doing electrocatalysis, we are placing only in electroactive material on the electrode, and then we add different concentrations of the analyte to actually monitor the signal. So in this case, we were doing some detection of ethynyl estradiol in water. And we have found that now the signature or the signal, because after synthesizing all this material, we need to look at their functions before including the analyte. And now when it comes to the wastewater detection, we have found that, yes, this is water without the analyte. So after adding the sample of EE2, which is ethynyl estradiol, we can see that the signature started coming out, and it actually appears in the same region as our normal sensor in buffer solution. So it is very important to test your sensor in wastewater bodies. And we have calculated our limit of detection, the sensitivity, and the dynamic linear range. We have compared our sensor with the previous methods, and we have found that, you see, this work has a low limit of detection compared to the other methods. Even the linear range is smaller. You can see that it's from 0.12. So it can detect very low concentrations of these contaminants in water. So now in conclusion, the biosensor systems can be used to detect various analytes in water. So what is important there is that we need to check so many things before applying our sensor system in water. And there is a possibility for replacing other technologies or modifying other technologies with the biosensor. As you have seen that the biosensors obtained high sensitivity, specificity, and better limit of detection. So these are my references. Thank you very much.